First of all, I would like to say that it's a great pleasure to join you here in this meeting, 6 years after my first Human Glycome project meeting in Dubrovnik, and also thank Gordon for this excellent program, as usual, for the Human Glycome project. And today I will show you one of the projects that is being developed at the Glycobiology in Cancer group at I3S University of Porto. And this presentation will be focused on the ST6-Gal1 role in modulation of antibody therapy response and immune evasion of colorectal cancer cells. And as you have heard during this day, glycans are key components of the biological systems, and they are involved in several mechanisms like extracellular communication, signaling, immune modulation, recognition of different pathogens. And the glycosylation process in the normal mammalian cells is tightly regulated. However, in cancer cells and during malignant cell transformation, this biosynthesis of glycans is altered, and we have altered expression of glycosyltransferases and mislocalization of them also. And they will lead to the expression of aberrant tumor-associated glycans. And in our group, at the Glycobiology and Cancer group at I3S, we mainly use two different cancer models, gastric or stomach cancer and colorectal cancer. And we analyze different biological samples, and using combinatorial approach, we aim to study glycans and their role in different molecular mechanisms as biomarkers for patient stratification and also as therapeutic targets. And in this presentation, I will focus in one of the enzymes that is being discussed here today, the ST6-Gal1 glycosyltransferase. And we know that in cancer, they are overexpressed, and they express alpha-2,6 linked sialylated glycans. And they mediate different signaling pathways in cancers that lead to tumor growth and tumor progression, but also drug resistance and immune invasion. And for sake of time, I will focus my presentation in colorectal cancer, the third most prevalent cancer and the second leading cause of cancer-related death. And although high-income countries already adopt different strategies to detect this cancer at early stages, they are usually diagnosed at advanced stages where we have limited therapeutic strategies. And of course, the patient has poor prognosis. So besides the immunotherapy and the different checkpoints that are used at that time in the clinic for patients with mismatch repair deficient status, we have this specific target therapy that is used for metastatic colorectal cancer targeting receptor tyrosine kinase. And one of these receptors that is targeted is EGFR, and it's targeted by a monoclonal antibody cetuximab or also panitumumab. However, only 10 to 20% of these patients respond to therapy. And we know that aberrant glycosylation has implications in anti-cancer target therapeutic strategies, not only in receptor tyrosine kinase therapies, but also immune checkpoint blockade. And so for that we decide to, and we are pursuing to understand the role of alpha-2,6 sialic acid in the establishment of malignant phenotype in these gastrointestinal cancer cells. And today I will show you three different parts of the work that is being done in colorectal cancer, and mainly the cancer cell resistance to anti-EGFR monoclonal antibody treatment, also the immunomodulatory role of this ST6-Gal1, and finally I will discuss a little bit the ongoing work with the functional impact of ST6-Gal1 in cancer cell-derived extracellular vesicles. So to answer this question, we developed at the lab two different glycoengineered colorectal cancer cell models. One of them, we use one cell line that do not express sialic acids, and we overexpress ST6-Gal1 in this cell line. And another cell model where we knock out by CRISPR-Cas9 the ST6-Gal1. And we analyze the glycan content of these different cells, and we saw that ST6-Gal1 expression reshapes the cell surface glycosylation of colorectal cancer cells. As you can see here, when we don't have ST6-Gal1 in CACO2 cells, we don't have sialic acid, and they are reshaping by terminal fucosylated structures, and the other way around in the other cell line. Moreover, we did a glycoproteomic of EGFR receptor, and it was really interesting to see that the cells that express ST6-Gal1, the glycosylation sites are specifically in the domain where the cetuximab, the anti-EGFR monoclonal therapy, binds. And then we treat these cells that overexpress ST6-Gal1 with cetuximab, and we observed that this modulates the susceptibility of colorectal cancer cells to monoclonal antibody target therapy with a higher metabolic activity, decrease in cell death, and downregulation of EGFR at the cell surface. So we know that ST6-Gal1 overexpression interferes with the cytotoxic effect of this anti-EGFR monoclonal antibody. Moreover, we show that ST6-Gal1 is present in colorectal cancer patients, and when we correlate this expression with the clinical pathological data of the patients, we observe that they are more expressed in patients in the MSS status patient, and also in the left side column, that is where these MSS tumors arise. And knowing this, we decided to understand better the immunomodulatory role of ST6-Gal1 in tumor immune invasion mechanisms. And as we heard today, we know that these glycan changes in the cells, the tumor cells, are recognized by glycan binding proteins, and that are present in the immune system, in the immune cells, and they have different affinities. And in this specific project, we focus in the Siglec family, that are the ones that bind specifically to sialylated glycans with different linkage, and also are present in different immune cells. And at that time, we have a brilliant PhD student at the lab that was interested in this project, and she takes advantage of these glycoengineer cell models that we developed, and she screened for different Siglecs that has alpha to 6 affinity, and she showed that when the cells do not express sialic acid, they do not bind to Siglec 10, in contrast, when we have overexpression of ST6-Gal1, they bind better to Siglec 10. And we validate that this binding is specific for the sialic acid during an neuraminidase treatment of these cells, and corroborating this further with different cell lines from colorectal cancer, and we observed that was a significant correlation between the expression of ST6-Gal1 and the Siglec 10 binding. And knowing that Siglec 10 is expressed in different cells of the immune system, we did a bioinformatic analysis taking advantage of a RNA-seq data set, and we observed that comparing the adjacent normal tissue with tumor tissue, Siglec 10 is expressed in a cluster of myeloid cells. We also observed this in tumor tissues, and also when we isolate monocytes and differentiate them in macrophages, we observed that they express Siglec 10. And when we went in detail to this cluster of myeloid cells and the different subpopulation, we observed that most of the cells that were overexpressed are involved in the phagocytosis. So we decided to perform a phagocytosis assay, a flow-based phagocytosis assay, and we observed that the cells that don't have, or the cells that do not have ST6-Gal1 are more phagocyted than the ones that do not have. and we validate this in an imaging phagocytosis assay, where we dye the cells with a specific, we label the cells with a specific dye that was only appearing when the cells enter into the macrophages and went to the phagosomes to be destroyed, and this red signal appears. So we corroborate that the cells that do not express ST6-Gal1 are more phagocytes, showing that ST6-Gal1 abrogates macrophage-mediated phagocytosis in colorectal cancer cells. And to show that this phagocytosis mechanism is dependent on the Siglec-10 expression, we knock out Siglec-10, and we were able to do this in macrophages, and we decreased the expression of Siglec-10, and we showed that when we decrease this Siglec-10 expression in macrophages, we have an increase in phagocytosis. So knowing that the cells also use some extracellular vesicles to modulate this immune modulatory role, we decide to study the cancer cell-derived extracellular vesicles that are released by these cancer cells. And I just want to let you know that these extracellular vesicles are nano-sized particles, and the most well-known are exosomes, and they have like 50 to 150 nanometers, and they are released by the cells to the extracellular component, and they are glycosylated. And our group already showed that there are different methodologies that are really important to isolate these extracellular vesicles, and they will lead to different glycosylation patterns according to these different methodologies, and that there are also specific glycans and glycosylation changes that are responsible and important for organ tropism, and also that the specific protein clusters of cells in this specific protein signatures in these extracellular vesicles have clinical impact in the prognosis of the patients. So what we are now doing is isolating these extracellular vesicles from the glycoengineer models and characterize them through the ISEV guidelines, through transmission electron microscopy, and also using some canonical EV markers. And at that time, we were able to show that the EVs that were isolated from the cells that do not express alpha-2,6 cellulation also do not express alpha-2,6 cellulation in these EVs. And we are now pursuing with a PhD student the glycomic analysis of these EVs together with Manfred and Norche, and also evaluating the effect of this colorectal cancer EV glycosylation in macrophages uptake. And I hope that I will be able to show you this in the next meeting. So in summary, in this presentation, I show you that the tumor-associated ST6-Gal1 cellulates EGFR and target EGFR in specific cetuximab binding domain sites, and that the terminal alpha-2,6 EGFRs interfere with the cytotoxic effect of the monoclonal antibody in colorectal cancer cells, and that ST6-Gal1 generates cellulated glycans of the Siglec-10 in colorectal cancer, and macrophage-mediated phagocytosis is also regulated by this ST6-Gal1 Siglec-10 axis in colorectal cancer. And we are now pursuing with these tumor-derived EV glycosylation signatures and the modulation of this in the immune response. So finally, I would like to thank to the Glycobiology in Cancer group led by Professor Celso Reis and the people involved in this work, mainly Álvaro, Joana, and Henrique, and also our international collaborators that were crucial for this work in the glycoimmunology part, Sandra van Vliet from Amsterdam UMC, and in the glycomics and glycoproteomics, the CPM group, and Manfred, Paul, and Norche, and the funding agency. Thank you all.
Thank you very much, Joana. Really great talk. Sure, there are a lot of questions. René?
Thank you for a nice presentation. I was wondering, so the extracellular vesicles,
Yeah.
why do you think these are important? Because you don't need them in the model, is it? You could also directly interact with the macrophages and thereby inhibit the macrophage.
So they are really important because they are released by the tumor cells, and they are important also for the cellular communication and the tropism of metastasis and of these cells. And we know that there are some studies that are showing that they have immunomodulatory role, and of course it could be through the macrophage uptake or others. We are still.
Yeah, I understand. Is there evidence they play a role in vivo?
Sorry?
Is there evidence they play a role in vivo?
I really don't know if you said if it's play a role in
vivo, in
vivo, yeah, in vivo. Okay, so it's true we are doing some experiments in the group, and this one that I show you, that the specific glycosylation partners are involved in this tropism of the extracellular vesicles were done in vivo, and we are now doing also in the lab that are isolating these extracellular vesicles from plasma of patients and doing this characterization. We also want to do co-culture with the immune cells of these patients, where we isolate the plasma and see that if this uptake of the macrophages is occurring also in vivo.
Thank you.
Perhaps a similar kind of a comment from myself, because it has been long known, old trick of the blood banks is to actually get rid of the glycocalyx of red cells to allow them to agglutinate, and that's because they think it's because of the negative charge they repulse each other. So, and people have also shown in various functional systems like ADCC and other phagocytosis systems that having this charge interaction is really negatively affecting. So I guess it could be possible that it's really as simple as simply a charge set, but it's probably also more complicated. I think so too.
Okay,
so other questions?
So very interesting talk.
Thank you.
So you mentioned macrophage-mediated phagocytosis is regulated by this ST6L1 and the Siglec-10, right? So I'm wondering, is there any possible mechanism why they can do that? Is there any receptor-mediated phagocytosis? For example, ST6L can bind to Siglec-10?
Yeah, we think that this glycoimmune axis will be really important as a novel target therapy, because we know that the immune checkpoint inhibitors is not efficient for all these patients. So we are also trying to understand if this ST6L1 expression in cancer cells is specific in specific proteins of the cells, or if it's quite randomly, and this axis could be interfering with this phagocytosis and, of course, in the immune modulation of the tumors.
Theoretically, this enzyme, of course, they can bind it to maybe the product cyanic acid, right?
Yeah.
So maybe this one can interrupt with ST6L with Siglec-10. Cyanic acid will bind that.
Yeah, we are also doing some work with inhibitors of this Siglec to show that this axis is really crucial and could be used as a therapy immune checkpoint. Okay.
Thank you.
Thank you.
Hi, lovely talk. I'm just wondering about the purification of your EVs.
Okay, so what we developed several years ago in the group, and this work was led by Ana Magalhães in the group. So we observed that taking in consideration the different methodologies that were applied for the people that study the EVs, we have different glycan signatures. So we optimized what was the best method to isolate these EVs and keep the glycosylation in these nano-sized particles, these AVs. So this was what we performed several years ago in the group. So because there are different types and methodologies using ultracentrifugation, using kits, using size exclusion, chromatography, and these different types of methods could really change a little bit the glycosylation that we have in the EVs. So it's important for us because most of the people that works with EVs are not aware about the glycosylation or they just don't care about it. And it's really important to know that the method that we are using to isolate will keep the glyco signature that we have in the cells, okay? And we know that these EVs also carry glycans.
Over here. So
Okay,
the EGFR is also heavily phosphorylated.
Yeah.
And you mentioned that it's heavily glycosylated. So is there some sort of crosstalk here? Do you believe that the mechanism in place?
Yeah, it could be. The point here with the EGFR is that we know that this glycosylation could also interfere with antibody binding. And this is really important. And as you may know, in colorectal cancer, the way that you are or not eligible for this anti-EGFR monoclonal antibody therapy is just if you have RAS mutation or not. So if you are wild type RAS, you are eligible for this therapy, and they don't take care. They don't take in consideration the phosphorylation of the receptor or even the glycosylation. So there are also in the industry a lot of concerns regarding this, and we are thinking that glycosylation could have a role, and phosphorylation. We were not studying this in detail, but it's really important also to check if it could be a crosstalk also.
Thank you.
Okay, thank you very much, Joana. Let's thank her once again. And then next up is Nika. And because I got a short course on what all the umlauts mean and how you should pronounce them, which I completely forgot. And I see that there are several in her last name. So please, could you pronounce your last name?
Nika Simicic.
Thank you very much. And the floor is yours.